everyone, I'm just in the continuing theme of little video tutorials on various things that you need to do for the various assignments and projects in the lab class. I have one where we're going to go ahead and we're going to download sequences for assignment 1 and probably assignment 2. What I have here is on the page here, there's the yeast genome database, Saccharomyces database. It's got a lot of information in it. You might want to just kind of look around at it. But the gene we're most interested in right now is one called PHO13. I'll probably call it FO13 in future videos. So I'll click on this FO13. And what it does is it shows there's a database of just general information, and here's all these little tabs that show the kinds of information there are, but we'll scroll down and look. It's the standard name and the systematic name and a description of what it does. It's in the phosphate metabolism group. It has some comparative information here. If you're interested in where it's located on the yeast, which chromosome in yeast, it's in chromosome 4 of yeast. Here's the open reading frame here, and some things you can do to analyze the sequence and things like that. Some of the alleles, the deletions. If you see a delta after a gene name, that means it's been deleted. And in fact, you're going to see that quite regularly in the rest of the semester because we'll be using a FO8 delta strain. It doesn't express another alkaline phosphatase in yeast. So the gene ontology, the phenotype, all kinds of stuff like this. There's all kinds of general information. If you want to know a little bit more about it, and a future assignment, if you want to find out more literature, here's some references you might want to use to start from for your reading a paper. But what we want to do here is to go to sequence here and download. And we're first going to go ahead and download the protein sequence. And I've already done it once before. And here I just go ahead and load it in. What I did is I created a folder called Bio4242 sequences, and I'm going to put that sequence in there. I'm just going to copy over. So I've already done this before. And what I'm going to also do now is in the future for another assignment, I'm going to have you use the coding DNA as well. So we may as well download that now while we're here. Here's the coding DNA. We'll save that as well. So now in a folder you have the two files, the protein sequence and then the DNA sequence of the coding region of the gene. If you need, for example, the genomic DNA or the DNA sequence, including 1 kilobase of coding front and rear, and this custom retrieval I think allows you to collect data in a different way. But let's go ahead and let that's one of the first things you need to do. Part of assignment number 1 is we want you to be introduced to Protein Blast. And so what I do is I recommend what I did here was I just typed BLAST and it just BLAST protein, standard Protein Blast. Go here. And you can get it. So not a different ways there. There's a link to it in Blackboard as well. We'll go ahead and just Protein Blast. And since we put the sequence in, we have that file downloaded, I can just click choose this file. This is in BLAST. Here's the protein. I'll click open. So it'll open that file. It'll automatically read these FASTA formatted files. What I want you to do is I want you to search the human. So type in human. If you notice the text is 9606, doesn't matter which if it's human or humans. Go ahead and click that because we want to only search the database for human analogs. So we potentially want to try to make links between yeast proteins and human proteins for this project. And then click BLAST. And what it's going to do is it's automatically go to the database. It's going to search and find related proteins. And what we want to do is we want to find a human protein that is closely related to alkaline phosphatase in yeast. And here we have a search. The results have come in. So here, the best alignment is this glycerol 3-phosphate phosphatase from Homo sapiens. Maximum score is 169, total score 169, percent identity is 33. So 1 out of every 3 residues is completely conserved. And this is the one we're potentially most interested in. So we'll go ahead and click on this search. And here we have an alignment of the sequences. So it's all the single letter codes, and it's only a part of the sequence. But you see that there's quite a few conserved residues here where the letters are repeated in between the 2 sequences or where there's identities. And if there's a plus sign, that means they're closely related, very conserved changes. So what I'm going to do is, since this is, I want to go ahead and get that sequence, I will go ahead and click on the sequence ID, click it right there. And what it's going to do is the databases are also linked where you can just find other information pretty easily. So here, glycerol 3-phosphate phosphatase from humans. And the NCBI, this is still the NCBI website. These are the resources. So you can go through, you can see the sequence, you can have references to the sequence information. You can go through here and you can find out all kinds of things. This is when I first start working on a new protein or things like that, what I will always do is I will go ahead and do a search like this just to find references, find out a little bit about the enzyme. Here's the sequence of it, and that sort of thing. But what I'm going to do is it's something we'll need to do is we're going to go ahead and do a FASTA. And what it's going to do is it's going to create and show me the FASTA file for this protein sequence. So I will highlight that whole thing, do Control C or Command C if you're on a Macintosh. And then what I'm going to do is I'm going to copy this sequence as it is into Notepad. And what I'll do here, just so that I know what it is, I will type human PGP and file, I will save it as human PGP. And now I have that. So now we've downloaded and created files for all of the sequences that you'll need to use for assignment 1. And these will all be sequences you're going to be seeing again in assignment 2 as well. So hopefully this helps you. If you have any questions, feel free to send me an email. But this hopefully will get you started with sequences, and I'll create another video of sort of more detailed aspects of assignment 1. In assignment 1, we're going to be doing another sort of an alignment of these 2 proteins. Then we'll get that'll set up, and we'll create a figure showing the alignment of the DNA and the protein sequence and that sort of thing. So hopefully this makes sense and this is useful to you, and we will see you later. Bye.